So step 3 is going to be contraction. Contraction, before we get there, let's just make sure we have all these terms down. Actin is going to be the thin filaments. Myosin is going to be the thick filaments. They're going to crawl on each other to sort of condense the muscle layer. Remember, these two don't shrink, but what they do is they crawl on each other to make that space thinner. Sarcomere, that's the area that they sort of inhabit, and that's going to condense. That's where the actin and myosin sort of get shorter because they overlap. Tropomyosin, that's going to be the regulatory protein that covers those myosin heads, and the only way we can get them out of the way is to activate tropin. And when tropin has been activated with calcium, they sort of move out of the way, and that allows the tropomyosin to move out of the way, of course, and that's going to get us to have these openings, which is going to allow the thick filaments, the myosin, to crawl over them. So let's kind of go over the process. Calcium binds to tropinin, and tropin then goes ahead and moves. So we have a calcium binding site right over here. When calcium binds to tropin, you can see the tropomyosin is covering these little openings. So calcium binds, they move out of the way. Can you see how they condensed? And before, we couldn't see these active sites where the thick filament can sort of bind and sort of crawl across. And now that the tropomyosin is out of the way, we can see them. And number 2, myosin can actually go ahead and land in these little pockets and crawl. We're going to actually see that in the next couple of slides. So let's go ahead and assume that calcium came in and attached to the tropin, moving the tropomyosin, so you can see that they're out of the way. And we know this because those active sites, those little holes, those openings, are available. And this is also, of course, the thin filament. This is going to be the thick filament. You can tell it's a thick filament because it's got that head on it. It's called the myosin head. And the myosin head attaches to the actin in these little openings. Well, the thing that makes the myosin head activated is by having ADP plus a phosphate attached to it. So recall ATP is adenine triphosphate. One of those phosphates fall off. It becomes ADP diphosphate. So the ADP plus the phosphate attach to the myosin head. And then what happens is the myosin head attaches to those active sites and moves a little bit. So let's say we moved in one direction. What then happens is we have to move this myosin head again. So we might be going from one spot to the next spot to the next spot to the next spot, moving these thin and thick filaments across each other so we can move that muscle from being extended to sort of contracting it. So how we do our next step is, so before I even get there, this is called the myosin cross bridge because there's now a bridge from the myosin to the actin, thin filament, thick filament. We have to go ahead and you'll see that now that the ADP has moved on that head, we have the stroke that occurred. So it went from this kind of position to this kind of position because of that ATP ADP. But once it does that move, it basically breaks off the adenine diphosphate with the phosphate. They fall off. And the only way we can get this myosin head to move again is to attach ATP triphosphate to it. So let's say we have to contract this muscle more and more and more. All we're going to be doing is going ahead and having this ATP turn into ADP plus a phosphate, move that myosin, it'll create that myosin bridge, so the myosin head will attach to those openings. The ADP and phosphate will fall off, That's going to go ahead and create that power stroke. And then to get that myosin head off of the thin filament, we're going to have to hit it with an ATP again. And then we can do this over and over and over again. If you think about your arm completely stretched out, and then you're going to slowly bring your arm in to sort of touch your nose, you're basically making ADP plus a phosphate do this reaction over and over and over again until you basically condense that muscle so you can sort of touch your nose or do whatever. So the ATP in the myosin head basically reactivates, and the ATP that was there gets broken down again, and it allows for the myosin head to do this reaction over and over and over. In our next video, we'll talk about contraction.